over, like as Kevin said, wildfires have been a big problem for many of us that live in the West Coast, at least, California, Washington, Oregon. So what I'm gonna do is go over a quick study that we did in 2020. I know it's an opportunistic study, right? I can't design a forest fire to study this thing. So we took advantage of that unfortunate event. So we did a quick study on how to at least look at the impacts of those wildfires on bees. And then probiotics, I chose to talk a little bit because we were doing a couple of projects, but then I decided we still don't have full data. So I'll just go over some basics of probiotics. Maybe some of you try to use probiotics. I thought I'll give you some insights on that. But I sneaked in some quick study on the European fall group. I thought it's more relevant for at least Washington and Oregon, where we are seeing more of UAB. So I'll go over a few of those slides at the end. Okay, again, I think you saw all those people yesterday. Again, without their support, nothing is possible. So I'm not the only one running the show. So I really acknowledge the support of all my past and current lab members. Okay, disclaimer. As I said, it's an opportunistic study. We just took advantage of what we had. So we all saw this, right? In the news 2020, we had a huge issue with these forest fires and temperatures. I don't know how much you got here, but Corvallis, at least where I live, I think the max was 108 something, and then I believe Seattle got 115 degrees. Yeah, that was a record, right? So so again, I'm not talking about heat that much today, but I'm looking at how does fire or smoke impact bees. So I don't know, George, I think probably is outside somewhere. So this is George Hansen. Let me ask you this question. Has anyone been impacted with forest fires, like something like this bad in the last 2 years? Oh, okay. 3 years ago, yeah, okay. And has anyone seen like a lot of impact of smoke on your bees? At least if we don't have fire next door, but yeah, okay. Some of you are nodding head. So this is unfortunate. What we saw was so this is George, and see, this is only one apiary. He was, I don't know, maybe some 300 hives were impacted with this fire for him. But this is more towards the Cascades, like foothills areas, where some of these apiaries are. So you can see some of those eyes, these are all missing eyes, they're totally burned. So you just see, it's amazing, right, how the fire goes, right? Sometimes these were not touched that much, and then you see some of these eyes gone here. So it's been a drastic. So we use some of these hives for experiments as well. That's the reason I'm showing you this picture. So I may come back to this again at the end. Let's see if this video plays. This is another beekeeper from St. Queenie. He lives in the valley. So he sent me this video. So what you're seeing here is, this is not a night vision camera. This is during the daytime, around probably 1 o'clock or 2 o'clock. He shot this video, and see all the bees are just bearding out. And this, like you all know, right? Bees use polarized light, even if it's cloudy, or they can still see where the sun is, or the position of the sun. But unfortunately, I think some studies have shown, not bee studies, but some of the physics guys that study all this different matter, they have shown that polarized light is distorted if you have a lot of smoke and the fire like this. So that's a big impact as well. That's why your bees are struggling to forage, because they see this, they can't really see where the sun is. And that's why you see that bearding. The temperatures are still okay to fly, right? It's still warm. But unfortunately, they can't see and at least find where the sun is. And their olfactory cues are messed up to some degree as well. So the effects of smoke and fire, I think that's what is not very well understood. If you find some studies, let me know. I've tried hard to find, if I can find anything that has really done a comprehensive analysis to look at how smoke or fire impacts bees, but I couldn't find any. But they have looked at some of the insects like butterflies. I have seen a couple of other studies, but nothing on bees. So we thought this is a good opportunity to take advantage and look for at least the minimal things that we could look for in terms of impacts of fire and smoke. So before that, just for fun, I have a few things here for you to think about. So probably you read a lot of bee books, and probably the new beekeepers here, you have been reading books. So what do you read when you say there is a forest fire? People say, oh, bees will abscond, right? Have you heard of that before? But how many of you believe bees abscond when there is a forest fire? Tricky question. Okay, look, I don't have a good answer either. I'm trying to speculate based on what I have read or what there is in literature. So let's go through the hypothesis. So it said here, so honeybee colonies can escape fire by absconding. That's what at least some people believe. So then to justify this is the thing, right? They say, like, when you smoke your hives, what happens? We say bees, they engorge on the honey, they imbibe honey and then try to go in. That's why they are not very defensive. They are not using those alarm pheromones. So that's the understanding. That's what I'm saying here is imbibing of honey by bees in response to smoke or fire. I think this is true. I'm not contradicting this fact. So when you smoke, probably, yeah, people have shown, they fill their guts or the crop with the honey or nectar, whatever is there. And they are, that doesn't mean that they are ready to fly away from the hive. So that's what I'm saying. They do engorge, but then I don't think they're ready to go fly away from there. So there is a lack of evidence regarding absconding behavior in response to fire and smoke. That's what my hypothesis is here. So what is the alternate hypothesis? The engorging behavior or the behavior of ingesting that honey is to sustain the foraging dirt in the aftermath of fire. So they are doing it. If I'm a bee in a nest in a big tree and there is a lot of fire, I am imbibing just to be safe that if something goes wrong, there may not be any forage available in the next few weeks. And I still have to build the nest probably to bring in more resources. Maybe that's the thing happening. Again, this is food for thought. I don't just take me for sure, because I'm just giving you some hypothesis here. So engorging is to sustain the foraging dirt in the aftermath of fire. That's probably makes more sense. I'll give you more evidence to discard this hypothesis that bees escape fire, right? So let's talk about some supporting arguments for what I am saying here. So you have the honeybee queen, right? If you read your biology already and have attended meetings, what do people tell you? Like, the queen has to lose weight, right? When they have to swarm. And then we say the queen has been chased around in the hive. They are fed less amount of food so that they lose weight. Their ovaries are shrunk. Then they can fly easily. So that's pretty much known fact. I'm not contradicting that one. So what I'm saying here is but in this case, these bees didn't know, right? They don't predict fire. They don't know that tomorrow or day after there's gonna be a fire in this area. So how will they lose weight of the queen, right? The queen can't fly. So the fire comes and they can't fly. They're still gravid. That's what I mean by gravid means they're still, the ovaries are fully mature and they are still grown, and she's still pretty bulky. So she can't really fly. That's another thing that you can think of. And then, so they have to coordinate, right? They can't just absconding doesn't mean that there is a queen and a bunch of bees will just fly beautifully with her, right? If you understand the swarming process, it's a very complex process, right? Where the scout bees have to go find a permanent nest. Then there is a lot of orientation that happens, and then they swarm. So if there is a lot of smoke and fire, can they even coordinate in that smoky environment? So these are two things that contradict this hypothesis. Hope I was able to sell my idea here. Again, I don't know. This is just a logical response. Again, nothing to do with your beekeeping will not improve tomorrow in this, but I just thought I would give you something to think about. Because sometimes books can be wrong, what we have learned all these days.
There must be a difference between swarming and absconding, because it's two different words.
Could be, yeah, that's a great question. They could be very different, yeah. So we don't understand, but I'm just giving you another additional information to think about. That's what I said. This is not the best answer, but could be. If you come with a better explanation, yeah, please email me. So, yeah.
Was there any evidence of George's hives that didn't burn leaving?
See, he's already coming to the point with, I'm going to come back to that in a second. Yeah. So like Paul is already advanced, so I'll say, so that's another evidence, right? So I wanted to show you this first and then go to George's. So this is a wild hive. So I was giving a similar talk to Nevada State beekeepers this year, I think in February. So they came and he gave me these pictures. So this is from one of their beekeepers. So he lives in somewhere outside Reno or wherever. So they had a fire a year before or whatever. And you see, this is a hive. So he lives pretty close to this place. This is his property, I believe. Right? So you see this trees all charred, you see a lot of trees burned here on the background. And you see there are bees here. I don't know if you can see very well where my pointer is. So there is a hive here and it didn't abscond. So that's another evidence that bees don't abscond probably when there is a fire. They try to engorge on the honey and then they stay inside the hive. So this is a clear indication of that hypothesis that maybe bees don't abscond when there is fire, but they try to regurgitate honey as much as they can, and they just survive the fire instead of absconding. And same with the, let me go back here with the George's hives that. So you see these hives, right? If the absconding behavior is so common in response to fire, then you should see these hives around, right? Yeah, see it. George came on time. So I'm telling you, right, George?
I heard you talking about it.
So you missed the hypothesis of absconding behavior. We'll talk later. So I'm saying like if the absconding behavior is true in response to a fire, all these sites, George, yeah, maybe they'll not believe me, but you had bees in these sites, right? That survived. Yeah, see, they didn't abscond. So fire went real close to them, but those hives are still alive and they didn't abscond. So I gave you two scenarios: a fire in a feral hive in the woods, and then I am showing you George's hives here. So that tells that bees don't abscond for a fire, but they have this engorging behavior to sustain for the next few days until the forage becomes available for them.
But what about the hives in the picture on the right that burned?
Bees-
Those bees take off.
Yeah, unfortunately, those bees, yeah, they just disappeared. Along with the hive, they carried the hive as well. Okay, let me go back to where I stopped. Yeah, so this is the feral hive I talked about. So, okay, that's enough about the basic fun stuff, but I'll just talk more about what we did after the smoke. So, yeah, there is this last slide to just give you more sense on what could the smoke or fire do to your bees. So one hypothesis would be the respiratory system, right? The respiratory system is the one you can immediately see. They have all this, for the new beekeeper, they have all these spiracles here for exchange of oxygen and carbon dioxide. So they could be blocked with a lot of ash or anything that flies into their systems. So that's what I'm saying, it could block spiracles or trachea, impact on olfaction. Of course, you as a beekeeper may say, oh, I smoke my bees a lot. I don't think bees get impacted. Yeah, too much smoke. So you are impacting their olfaction, right? That's how the alarm pheromone is not being communicated. And that's how your bees are not perceiving the alarm pheromone cues, because they have an impact on olfactions. But we don't really understand with the fire smoke that happens in big number, in a big way. We don't know what kind of, I mean, we know probably they can't smell a lot of phytochemicals and other things they're looking for, but no one has really looked at that at this point. There might be a lot of physiological stresses like oxidative stress. I'm not going detail about that. And of course, as I said before, I showed you that video with all the night vision camera I was joking about. It's in the daytime, bees can't really fly. They are bearding outside the hives. That's because of impact on orientation and navigation. As I said before, the polarized light that comes in is distorted. So bees can't really figure out where the sun is at that point of time. So some studies have shown the heat can impact the queens in terms of their sperm viability. So we wanted to look at that aspect as well. That's why when we took George's queens to look at those aspects. And then the toxic material. So think about this, forest fire is not just that ash or it burns the trees, right? But then if there are homes that are being burned, so they have a lot of carpets, there is sofas, all this material, right? Think about that as well. That comes in terms of flying into your homes or wherever the ash is gonna come. So we wanted to look at those elements as well. Are there anything that fly or come to maybe miles and miles of these smoky areas and impact your bees? So that's what we wanted to look. But as I said before, in honeybees, there are no studies I could find. So there was one on butterflies that they have shown that the hay smoke impacts survival and development of butterflies. So these are the two main objectives. We wanted to look at the fire impacts on honeybees and then on queens as well in terms of those sperm viability that I mentioned before. So these are some OSU hives, and again, as I said, it's an opportunistic study. We didn't plan or anything to do. So I randomly went after this incident and took samples. So here The air quality wasn't as bad as in some of your areas here, but it was about 275 when I took samples of foragers. And I went in and took nurseries as well. We wanted to compare, maybe nurseries are not impacted that much, only the foragers that are outside are impacted more. So we wanted to do a difference or see a difference between nurseries and your foragers as well. And then from the same hives that we sampled, we also went back again in October when the smoke was all gone and the quality, air quality was a lot better, 35. This is the first time I had to put that smoke, the respirator on. So these are the samples that we collected from these different hives here. So we are looking at electron microscopy. So here we have a good electron microscopy facility at Oregon State University. So this is how we make the sample slides, and then they go into their scanning microscope, and then they provide us the results. If you have seen scanning electron microscope images before, they look something like this. This is not from the direct study, but it was another. I'm just showing you how clearly you can see if there are things. So this is actually a pollen grain, not a. So we are still doing this. So we haven't analyzed the data yet. So whatever I'm going to show is very preliminary today. So we are looking at this is the tray, the spherical that I talked about. You know, taken tubes are connected, right? So this is the opening that you see. We want to see if any particles are deposited here and blocking the sphericals. So this is another image again. This is a forager honeybee during wildfire smoke. This is a nurse bee. So we haven't analyzed entirely, but I'm seeing some particles here and there. So hopefully the next few months we'll have some time to really analyze it. So as I said, we are also looking for elemental analysis to see if there is any other elements, not just the sphericals that are blocked by the particulate matter, but we are also looking at the elemental analysis as well. Okay, so I have some results for this. I thought I'll focus more on this study where we looked at queen health. So George was gracious to give us 20 queens from the hives that were impacted, where the fire went through like this. And then we asked him to find if he could queens of the same genetics in another apiary that wasn't impacted, right? Because I can't randomly compare some queens. So we wanted similar genetics if possible. So he was able to find 20 of the other hives that were not in the fire zone. And then we looked at a lot of different things on the queens. So I'm sure you'll see here. So these are some things we look for. Morphometrics, nothing will change, but we wanted to look, right? Just a fire event will not change the weight or the thorax width or head width of the queen, but we wanted to look at those as well. These are the important ones we wanted to look at: sperm count and the sperm viability. So again, I don't do this at OSU, so we send it to David Charby, who does most of those things at North Carolina. And then we still have to do some proteomic analysis at OSU, which we haven't done yet. We skipped this because I think so. Is anyone interested in seeing how to dissect a spermatheca? No interest? Okay. So that's a queen. So we didn't see any differences in mass or thorax or head width. These are not significantly different. But here you can, we didn't see any difference even in the spermatheca, number of sperm. So we are not seeing any of, even in the viability issue of the live sperm, looking at those things, we didn't see anything. So these are all non-significant differences. Looks like the hypothesis what we have now here is maybe the queens are buffered so well in those hives, even the temperatures are so high outside, but the queens, probably the workers are making a good cover next to the queen, and probably they are protecting the queen from any damage to the sperm or something. So that's a good news, at least, that we didn't see any huge problems. But we still have to do the proteomics. Hope we'll see something there. But again, looking at just the queen health, we haven't seen any problems. Yeah, George.
So, I mean, to me, this is astonishing because queens that are shipped and damaged because they overheated.
Yeah. So
I mean, the heat that they're experiencing is nothing
like this. Yeah.
Nothing like this.
Yeah, I was also surprised and to see these results that we are not seeing. But that in that case, there are only a few bees, right? Here, this is like an entire hive that is probably protecting the bee.
The combs in those hives
Yeah.
did not melt. There were no.
Yeah.
No melting, so I mean, somehow they managed to.
Yeah, so it's kind of very surprising, and so, yeah, we are learning as well, so, yeah, I was pleasantly surprised that, oh, these queens were not impacted with that smoke, so looks like, yeah, I think they have a good buffering mechanism inside the hive that they can protect their queens really well, even if there is high temperatures outside for some time.
And these were fast moving fires.
Yeah, they're fast moving too, yeah, that's a good point too, so we don't know how long they were in that temperature as well. Okay, so yeah, this probably will yield something, so we don't know, this takes a little time, so we are still looking at any other proteomic changes in those samples that we collected, so that's the fire part. If you don't have any question, I'll switch gears and go to probiotics here.
Add one.
Yeah.
Is queen egg production influenced during the fire?
Yeah, that's a great question, but we didn't follow through those hives, right? Yeah, maybe I don't want any fires, but if something happens, we'll try to look for those in the future, but as I said, none of these was a design study. We're taking opportunity for whatever that existed to look at it. So yeah, good question. Who knows how they perform? So maybe next time, if there is a chance, we'll look at a long-term impact. So how many of you use probiotics? None?
I did
Okay.
once.
Two of you, okay. You used once and you stopped?
Yeah.
Okay, because you didn't see anything happen?
I didn't see anything.
How about you, Matthew?
Yeah, Olson tried it.
Nothing happened? Yeah, okay. So probably the others are all clever, not using and wasting your money then. I don't know, okay. So I don't have any results here. We have a couple of studies that we just finished, so I may present those results later sometime. But I'll just give you some food for thought because I know probiotics is a big topic. I get a lot of questions about probiotics. So at least I'll make a definitive conclusion based on what I know is there is no conclusive evidence at this time that probiotics are doing anything significant to your colony. So probably at this point, I would not recommend using until we get more data from other labs and there is more degree of confidence, then maybe you should use. But I'll give you or at least make you think about some limitations or where we are in terms of probiotics so that you can make your own decision whether you should use probiotics or not. So I'll go a little basics on like few slides and then I'll switch to the European call group in a second. So probably most of you know honeybees have this gut microbiome and most of the bacteria, the beneficial bacteria, the most species are from here on. So ileum, this is your foregut, right? And then this is your midgut. From here on, it's hindgut. So at least the studies that these people have done, Nancy Moran and others in University of Texas, and there are other labs that have done some work on honeybee microbiome. It's still a very nascent field, I would say. We still have to understand a lot in terms of microbiome of honeybee. The human microbiome took so many years. We still have not found everything that we need to know about human microbiome, but bees have been only studied for the last 15 years, I would say, since 2005, I believe. There has been more research on honeybee microbiome. So there are still so many things that we don't understand. But we have some basic understanding for sure. So most of the gut microbiome is here. And bees have about 8 core bacterial species, and there might be multiple species within that same genus sometimes. So that's what we know, and have some things for you to think about as well. On the next slide, I will come on this one. So this is another review paper I found recently where they are showing how the microbiome is useful. It protects from adult pathogens, helps in digestion. Everyone knows this part. Detoxification of food, and development of the bee. It's a very vague one. And then the immune stimulation as well. Because bees have a lot of pathogens, right? So there is this notion that all these beneficial bacteria that are in the guts could help somehow arrest or hinder the populations of these pathogens. And sometimes they also talk about flowers, right? When bees go in, they could get some pathogens from here, or they could also get some of their beneficial bacteria because the other bee might have either done some defecation there or something like that. So there is an exchange of those beneficial bacteria as well as some bad ones too, between the floral resources and the bees that bring it back. So that's basically what we understand in terms of the exchange of microbes. So these are a few things probably you already have thought about, or maybe if you didn't, now it's a chance for you to think more about it. So most of the honeybee bacteria requires anaerobic conditions, right? When I say anaerobic, all of you understand you don't need oxygen there, right? So that's how they have to grow. So those of you who have used the the probiotics, have you tried to read the label? They have a bunch, I know, even I don't understand. I'm not a microbiologist. I know some basic bacteria, but I don't know all of those species or genus. So I'll not name any. I know there are 4 or 5 big companies that are into probiotics. As a researcher, I'm not supposed to talk about companies. So I'll keep that anonymous and say, read the label next time for whatever you have used or plan to use. There are a bunch of bacteria they list, and there are several of them. They are aerobic. That means they need oxygen to grow. Whereas your gut, there is small amount of oxygen, but most of the gut bacteria in honeybees, they are anaerobic. So that means they can't grow in presence of oxygen. But most of these bacteria that you see in the list, maybe half of them, they need oxygen. So that itself tells there is something wrong. They're just putting a lot of things because they know about bacteria, because human probiotics is so famous. So maybe they have copied some of those bacteria and just included in the honeybee microbiome as well. So I haven't gotten a good answer from, I've called a couple of companies to understand their logic of having some additional bacteria which honeybees don't have. What's the purpose of those? So we still don't know what those bacteria do. If they don't do any damage, you're okay. If they do some damage, then that's not a good thing for your bees, right? So think about that. Again, I know none of you is using, except both of you use and you don't use anymore. But if you want to, I think I want you to wait until there is more information and use of probiotics for bees. Any questions? Okay. And then this is just information for you. So the newly emerged bees, when they come out, they have shown there is no gut bacteria for them. So they are probably literally clean. So if you give me some newly emerged bees from an incubator and they don't interact with any older bees in the highway, just keep them in a cage, probably they will never have the bacteria in their guts. So that's what it is. The newly emerged bees, they don't have any gut bacteria. And you may say, then how do they get their bacteria? So that's what at least a couple of studies have shown. It's probably the fecal oral route. So they can't just get everything from prophylaxis. If I'm a young bee comes out of my cell and I'm interacting with my older sisters, that's how I get some bacteria by prophylaxis. But then they have seen if they don't get any contact with the fecal material, then they still have some bacteria absent. So both the oral prophylaxis and the defecation, if the other bee somewhere defecates, if they get an exposure to that, they acquire the rest of the bacteria from there. So that's a known fact is now at least we understand that most of the bacteria, the gut bacteria, the beneficial ones, they acquire through that fecal oral route. So that's what, I think that's enough for you for today to understand about probiotics. I'm not going to show any results here, but I think I have some conclusions to make based on this recent paper I found at the same lab, the Nancy Moran at University of Texas. So I'll just read the important points here. They say no probiotic formulations have been demonstrated to be reliably effective in honeybees as of today. So that's what I said to you as well, right? So they further say currently available probiotic formulations for bees include strains of Lactobacillus bifidobacterium used in fermenting dairy products and incorporated into human probiotic formulations. So that's something I have concern is the companies that are existing, they have randomly chosen either some human probiotics as well and tried to include within the honeybee microbiotics as well, probiotics too, which I don't know if it's a good thing or a bad. So that's what they'll say. These are foreign to bee guts. Results presented here in the general surveys of bee gut microbiota indicate that these bacteria do not establish to high titers or persist in bee guts. So they have also shown that these random things that are there in the probiotic mix, they don't grow inside your honeybee gut. But then they also contradict that and say potentially probiotics can provide benefits without establishing in the host, but robust evidence is not available. And this is the important one here. They say ingesting substantial quantities of live bacteria that do not occur naturally in your host, that means your honeybee, has the potential to do harm. So rigorous experiments that evaluate effects are needed. So that's where we are with probiotics. Hope that makes sense. And so maybe if you are very desperate to use probiotics, wait for another few years until there is more science and more conclusive evidence that they are doing some benefit to you. Okay, I'll. Okay, I still have 20 minutes.
Ramesh,
kind of.
If you had a problem with European foulbrood and used antibiotics, that would negate any probiotic application you had applied. Is that correct?
Yeah, and that's a good question. So all of you heard, right? So I know we all use antibiotics, especially for EFB these days, where there is a problem. So at least, George, what I have seen, again, I have not tried any of those in my experiments yet. But at least a couple of studies that I am aware of, they at least have shown to some degree, or they speculate that antibiotics will have an impact on the gut, the microbes that are there in the honeybee gut to some degree, but they haven't shown which one will be totally impacted or not. They are speculating at this time. So again, not much information by
The
feeding.
directions of the product that I bought, in fact, pointed out that you,
Okay.
you know, if you use this and then you
Yeah.
use antibiotics, it will negate it.
Yeah, and it probably depends on the antibiotic as well, right? It's a very broad spectrum. Maybe it kills everything, cleans the gut totally, maybe. But then those bees, I think if not every bee has consumed, maybe there is a way to acquire them back from the other bees after some time, right? So there is still a scope of acquiring, but it might impact temporarily for sure. Any other questions? Yes.
So I know in some other forms of livestock, people have done studies using prebiotics instead of probiotics. No, if anyone's done that for bees or has thought about doing that.
Yeah, no, that's a good question. Yeah, I have seen some prebiotics one, and they have shown some promise. Actually, the same authors, I don't have it top of my, just Google for Nancy Moran, UT Austin, and then click on prebiotics. They have tested some, I believe. And they have shown some promise, at least with one or two prebiotics I've seen. They have shown that, or maybe it might be a different author, but it's easy to find. Just look for. But I'm aware that people have tested prebiotics. Okay, this is something plug for me for seeking some help from you as citizen scientist. So I didn't plan on talking about this. I included this last night so that I can see if some of you are interested in helping with this project. So we have a USDA grant we got like a year and a half ago to create a pollen database, pollen composition database, nutritional composition. So I'll give you some context and then I'll ask your help on collecting some pollen for us if possible. So there is a lot of push, right? I know in Washington, you go to probably anywhere from Seattle to any rural areas. Probably there is a lot of push for developing habitat for bees. You see a lot of signs like this is in Portland somewhere. Then this is something we have done, tried to do. Not much has been done, but so I work very closely with the carrot seed industry in Madras, Central Oregon, and so their bees don't do well. Many beekeepers are reluctant to go there because it's a hybrid crop, doesn't produce enough pollen and nectar. Bees just go down within a month. The queen stops laying eggs literally sometimes. So we have tried to push for this, like you see some bachelor's button rows, hedgerows to plant it. This is carrots in the background. We wanted them to plant. So yeah, it's still not gone too far. We have been trying new farmers every year to convince, and some of them are convinced, but then water is a big issue there too, not just in California almonds. Even the carrot industry is finding enough water to produce carrots now because they didn't get much snowfall in the last 4 years or rain either. So it's a struggle, but again, that's a different topic. I'm not going to distract myself there, but so I'm just saying there is a lot of push for these type of things so that our bees have a better foraging habitat. Like Pam has the Seeds for Bees program. It's a great program. I don't know, George, they have distributed 1% land is under. Is that what the correct number?
It's like 50,000 acres.
Yeah, so it probably, they say 1 or 2% of the total acreage maybe, yeah. It's still small, but that's how you try to grow, right? So I think there are nonprofits like PAM that have done a great job of promoting the same concept of improving forage for bees. So again, based on this context, it's all great. So our project is not about that technically, the one I'm talking about. It's about developing a pollen nutritional composition database. So here we are asking citizen scientists like you to help us with pollen collection because we have been pretty ambitious. I think, committed for about 150 different plant species that are attractive to bees or used by bees. So we want to collect that pollen for analysis like in a comprehensive way. So you may say, why are you doing this? And so I'll just give you a justification here with some French fries. So before that, let me go to the next slide. So you see all this beautiful list, right? This is a USDA plants database where they have this beautiful list of plants that as a backyard beekeeper or as a commercial beekeeper convincing a farmer or someone to have some habitat. And probably you as a beekeeper get questions, or maybe you are curious, right? You may ask me, give me top 5 plants or top 10 plants that I can plant in my backyard that are good for bees. I can throw some 5 plants, but it's not a very robust list that we have right now, because most of it is based on attractiveness of the plant, right? If I stand next to lavender and I keep counting bees in a minute, they say, oh, I got 100 bees in a minute, this is very attractive. But I don't know the composition of pollen, I don't know the composition of nectar in terms of concentration of sugars or anything. I'm just giving you because it's attractive, I know bees like it, right? Anyone contradicts here? Is that how we have, right? It's all based on attractiveness. I have not seen anyone telling me, I mean, there are studies that people have done, they'll say, oh, lavender has this much pollen, pollen composition has this much crude protein, maybe very basic information, very scattered. So the whole idea of what, even like, let me go one more slide down. So like Xerces Society in Portland, they also have a beautiful list and people use those for planting things. But again, these all are based on attractiveness. That's what I wanted to make the case. So coming back to this slide, I want you to think, does attractiveness always translate or equate to best nutrition? So that's why my French fries come in. So you put salad and French fries, I may start eating French fries a little bit first because they look tasty, right? So again, I think Becca will be laughing. I told the same joke to their group recently. And so I think I was talking to some California state beekeeper or someone. I asked that, oh, am I not gonna eat French fries first and because they taste good? Then he says, no, it's because they get cold. So that's why you will eat French fries first. Okay, that's a good joke. I'll use it later. So here, what I'm saying here is that if you put these two together, it's a lame example, I guess, but still, I just wanted to drive my point through here. So when you have these two things here, this is the most healthy one probably when compared to this. This is just going to give me some carbohydrates. So when you say that put this thing and because as bees go to lavender, I may go to this first. Doesn't mean that this is attractive. Because this is attractive, this is the best thing for me. So that's the whole case. Most of these plants, that's what if you can read the bottom line here, the current basis of selection of plants for bees is based on attractiveness or bee visitation, not based on scientific information on the pollen composition or the nectar composition. So that's why we want to do this study. So yeah, let me go back here. So NRCS is really excited. Like I get calls from them every month now. When are you getting all the data? It takes a long time. First thing is we have to collect pollen, then pollen composition happens, and then we get the data. So the whole idea is the NRCS or Xerces, these people will use is they want to use the data that we generate and put it next to that saying, again, I made up this column here for you. So it will say crude protein, amino acids, sterols, lipids, omega-3 fatty acids. There will be a lot of things we'll be looking in that pollen. So it will be a more comprehensive analysis, right? Then you go back as a beekeeper or a citizen scientist who wants to plant something in the backyard, and you go and say, oh, this looks like a good thing for my region. And then you can go, you pick like 4 or 5, and then you look at these ones and you say, oh, they don't look pretty promising. So let me go to another one. I think this will help you choose a better plant based on nutrition as well, not just attractiveness. So again, it looks like a simple concept, right? It's a very painful one. So we need a gram of pollen, and we have been struggling to get gram of pollen for each species. It takes a lot of flowers. And so one of my graduate students, he designed this vacuum here. I'll talk about this in a minute. But there are different flower structures. Some produce more pollen than the others. It's a pain, right? So that's why if you decide on a plant, just email me that this is there I have. Can I start collecting some pollen? So you have a 2 years time from now. So you can collect whatever you have, but I need a gram. So for some blueberries, we tried a twisting method. You see some pollen is coming through here. I don't know if you can see in the back, but we had to design. No one had done this before. So we have all these techniques we are learning new. So we are trying to publish a paper on just the methods as well, how to collect pollen from different species of plants. So this is for blueberry. And for some, the bagging may work. You have those bags they sell for covering so that the bees don't go into the. So some of those can collect some pollen. And then is this swanication, because you know bumblebees, they do the buzz pollination. They have to swanicate to get the anthers burst and pollen come out. So we have tried to use some toothbrush, electric toothbrush on the bottom of the flower to get some. So again, these are very cumbersome or laborious things, but this is the one which might work for many species. So we try to design a vacuum, a handheld vacuum. So this is a backpack type that you put it here and then you see that. So I'm going to show you what this looks like here. So we designed all these different micron sized meshes here. So this is the 200, I believe. This is 58 and that's a 7. This is the smallest screen that you can think of. And we looked at based on the pollen grain size. So the graduate student, he read a lot of literature to find out the range of pollen from the smallest to the largest. So it looks like 200 to 7 is a good range. So then, so there is this attachments you see, he made those, you can buy those from wherever Lowe's or wherever you go, pretty hard. But then we had to find these exact meshes. And you see that's the external one is the largest one, 200, then comes your 58 and the least one is here with the 7. So if the pollen grain is too small, it will go all the way and deposit here on this screen inside. And then we meticulously have to remove that from that screen. So it takes a lot of effort. For the maple was the easiest one. We collected some maple this year. I think 2 students within 45 minutes they were done with the vacuum. But there are other species, it's painful. We collected for days and we still haven't gotten a gram. So again, if you want to watch, there is a video link here. I will send you a PDF of this if you're interested. Just email me. And so there we have discussed all these different methods, how you can collect pollen and provide us some samples. But if you think this is all a painful exercise, you don't want to do any of this, then I'll show you how to collect from bees, not from pollen traps. Pollen traps get a lot of different things. It's a contaminated pollen with different species. We want at least 90% pure pollen. So I'm going to show you how to collect bee pollen from their legs directly. So yeah, actually I have a video here. Let me. Yes, please watch this. I think this is very much self-descriptive one. There's a cool music in the background, but I don't think it's connected to the mic here. That's okay. We need about 50 to 60 bees if they're getting a good pollen load on their legs. So that would amount to a gram. So this is for people who don't want to kill the bees. I know some people say, oh, I don't want to kill the bees. So then you have to freeze them for 2 minutes, make them sleep, and then you take them out.
It doesn't matter that you kill
You know, it doesn't matter. I kill bees all the time, right? As a researcher. So for those, I know we wanted to accommodate everyone's needs. So yeah, you can kill them in the freezer, right? You can't put alcohol or anything because the pollen will get damaged. So. So once you collect, you have to put it in the freezer because the pollen loses its quality in terms of protein and other nutrients in there. So you put in your test freezer or -20 in your regular freezer, and then you have to ship them either overnight or in dry ice if you're going to take a little more time. Any questions on that? That's my email. So this tells you how obsessed we were. Brandon and Dr. Kiran and I went yesterday because Brandon said like a mile away from here there is this hemp. I'm interested in hemp pollen as well, but unfortunately we didn't see any bees there yet. So while going back, Dr. Kiran and I think John wanted to come as well. So we'll go and try some more pollen today. So this shows you my desperation that I need a lot of pollen. So if you can help, that would be really great. Yeah, John.
Have you considered using an electrostatic charge to collect pollen? I've seen some articles in pollination that this is for use charge to collect and dispense.
Yeah, that's a great question. So he's asking about electrostatic charge for this. Yeah, I read some papers, but I didn't see any real good protocols on if it will work for all these different species we are looking for. So yeah, if you have any ideas, let me know. We are still very challenged. I know about the idea, but I haven't tried it, so how to design it, and I don't have the engineering brain yet. So yeah.
I don't know, some of the work that being done by me and by have had fun with others who and Clint Otto, in North Dakota, indicates that, attractiveness is not just like attractiveness for like, it turns out that the honeybees go and here, and even though there are other plants and other kinds of bees go to that, so I mean, it's different kinds of attractiveness, and there's almost no overlap. And so, do you have a list of, or how are you sorting that out, or is it important that this one is great and this for all the qualities of the pollen, but it's only visited by this kind of bee?
Yeah, no, that's an excellent question, actually. So hope others understood, like, there are other people like in North Dakota, there's a USGS folks, and so they are all trying some different things. And so George is asking, how would you, so we have, that's why the 150 species, it's a very comprehensive list I have. It covers like most of the agricultural crops that our bees pollinate. And we have looked at other sets, so we have scoured through the literature to find wherever the attractiveness is a factor as well, and the bee visitations, and we collected a lot of different angles. So I'll send or share with the list with you if you think there is something missing. So I think we have covered at least 90% of the important plants that are available for bees as a forage.
When you say bees, you're talking about honeybees?
Honeybees, I'm talking about honeybees here. Or in that case, like if someone tells me like rabbit brush or something, I'm interested in that as well. If any, like, yeah, maybe I should correct myself. Initially, we thought of honeybees, but because we'll be restricting a lot of plants that probably honeybees are only specialized in going, or so we made it a more broader one. If someone sees some native bees crazily foraging on something, we want that pollen. Because honeybees are more generalists, probably they're not visiting for a reason, as you said. They have this beautiful behavior of directing their sisters for a more economical resource, probably. But bumblebees and all, they don't have that similar behavior. So yeah, we are including everything that bees are attracted to, not just honeybees. Any other questions?
Can you go back a couple to get your email address? I think I have it, but
Yeah, you can Google Oregon State, there is my email there too, on the bottom. Or if you want me to go up there, that's my email too. Are we getting close, Martina? That's what
Yeah, we got.
Oh, okay.
Yeah.
I'll wind
I
up
can't
in,
start them until you're
I
done.
will wind up in 3 4 minutes.
Thanks, guys. And then the graduate student showcase is gonna be in the other room, so everybody can move over there for that next one.
Cool, okay.
Thanks.
Okay, so, yeah, I just put this, this was funny, we were laughing. It says less than 0.3% THC cannot get you high.
You just have to smoke a lot of it.
Yeah. So yeah, it looks like I have only 3 minutes, so I'll quickly go over this EFB project. This is still an ongoing one. I don't have any big conclusive results here yet. But I know, I thought it's very relevant to Washington beekeepers, to at least the commercial guys that go for blueberry pollination and see a lot of EFB, but have other hobby beekeepers seen EFB in their hives recent this year in spring? Okay. So yeah, it's becoming, I know it's on and off some years, but again, it's more about food for thought for you, or what is resulting in your EFB incident. So there are a lot of hypotheses from nutritional stress. Is it a new variant of EFB? Is it fungicide exposure? Like I think Dr. Gao was showing about fungicides in blueberries. Is it fungicide exposure? Or there are some weird hypotheses about the pH of blueberries. I don't like this one, but it's there in the literature. The blueberry pollen, how many bees collect blueberry pollen? It's literally 1 to 2%. We did some studies, like Ellen, one of my students, when she was doing her master's with me, we trapped pollen from a lot of different crops where our beekeepers go. And blueberry pollen is only 1% of their total pollen collection that you look for. So blueberries don't produce enough pollen. I mean, bees don't like, basically it's a bell shape. Honeybees hate to go in and collect pollen on their legs. So blueberry growers may say, oh, why am I paying money to Matthew for putting bees in blueberries? The nectar foragers do most of the pollination. So the nectar forager, and we did that. We collected nectar foragers and quantified pollen on their heads and legs and other parts of the body. And there were at least 10 pollen grains on their bodies, which is enough for a blueberry flower to get pollination. So that's how, probably if you have questions from your blueberry grower, you can say the nectar foragers do the pollination, not the pollen foragers. So this hypothesis is totally discarded because this was like an 80s thesis from, I think, Gordon Wardell, where they said acidic pH of blueberry is doing it. When there is no blueberry pollen, where is the acidic pH going to come from? So I don't think that's why I shouldn't even put that on next time here, but I thought people think about it also, so you should know. Brood chilling could be another factor as well. So we're looking at all this different thing, I should wind up in a minute, so. So there is another thing between larval hypothesis, right? If you have too many nurse bees or too many older bees at a point of time, and so if suddenly there is a nectar flow and most of your bees become foragers and they're fine, so there'll be less nurse bees to take care of your larva, and that can create a larval nutritional stress as well. So you should think about colony demography as well when we talk about EFB. I know I have no time here, so I'm just skipping through. So we did a study, we were funded by Oregon Blueberry Commission to look into this problem. It's still an ongoing study, so I'm not going to show you very much results, but we have been trapping pollen as well to look for fungicides and insecticides. So we do a lot of colony evaluations as well, right? While looking for EFB symptoms. We collect if there are symptoms, we have these kits. We want to see if it's a different variant. So we have been collecting samples to look at the strain of EFB as well. And we did a quick study with Georgia sites looking at OTC resistance, but I'm not showing anything. But we didn't find any resistance for terramycin for EFB at this time. It's still working, so you can still use it. Okay, again, some dog pictures for Steve Shepherd, and I'll stop you.